Packed cell volume PCV. When anticoagulant blood is centrifuged at a standard speed, erythrocytes which are heavier than white cells and plasma will settle down at the bottom. This red cell volume is known as a hematocrit or packed cell volume. The hematocrit or packed cell volume is the volume of red cell expressed as a percentage of whole blood. There are two methods used for determination of hematocrit: macro hematocrit, micro hematocrit. Macro hematocrit, a large volume of blood is required in this method. Approximately 2 to 4 ml is required. Principle: anticoagulated blood is taken in ventral tube, filled up to the uppermost mark, and then rotated for a desired length of time. The packed cell volume PCV of a red cell is directly related from the graduated tube as a percentage. Requirement: the blood samples EDTA or double oxygenated anticoagulant blood is used in this method. Determine PCV within 6 hours of blood collection. The ventral tube, it is 110 millimeter length and 2.5 millimeter in diameter. The lower 100 millimeter are graduated or marked from 100 at the top and 0 at the bottom for PCV. The long-necked pasture pipette or special type of syringe is used for filling the ventral tube. Centrifuge machines will no speed. Procedure: mix 0.4 ml of EDTA with 2 ml of blood. Fill the ventral tube up to uppermost mark with the help of pasture pipette or a syringe. Fill the another ventral tube to balance the first one. If the blood sample is not available, fill the tube with water. Place the ventral tube in opposite side in centrifuge. Turn the centrifuge to slow speed, then slowly increase the speed to 3000 rpm. Centrifuge for 30 minutes at 3000 rpm. After 30 minutes, switch off the centrifuge and allow it to stop by itself. Take out the ventral tube and read PCV directly with the help of a graduate mark given at the tube. In a normal value, in male 42 to 50%, in female 36 to 38%. Micro hematocrit. This method requires a small amount of blood, 2 to 3 drops only. The blood can be obtained by finger puncture. Principle: the anticoagulated blood is centrifuged in a sealed capillary tube. Then PCV is determined by special hematocrit leader. Requirement: blood sample. Blood from the finger puncture may be used for EDTA or double oxalate. Venous blood can also be used. Capillary tube: use plain capillary tube for anticoagulated venous blood and use heparinous capillary tube coated with heparin internally. For blood obtained from finger puncture, the capillary tube is approximately 75 millimeter in length. Micro hematocrit centrifuge: this is a special type of centrifuge. It has a speed up to 15,000. The top of the centrifuge is flat with grooves. The centrifuge also have a timer which is usually set for 5 minutes. Hematocrit reader: there are several types of reader used for reading hematocrit. The simplest method is used of a card reader which can be made by hands. Clay: this is used to seal the end of the capillary tube. Procedure: draw the blood samples into appropriate capillary tube with the capillary action. Use plain tube for anticoagulated blood and heparinous tube for plain blood. In case of finger puncture, the blood should flow freely with the little puncture. Now wipe off the first drop and then collect the blood sample. Fill the tube about 3/4 length with blood. Seal the another end of the tube with clay or wax or ultimately by heating. The sealing should be about 2 millimeter depth. Place 2 HCT tube in the grooves of a centrifuge exactly opposite to each other. It is not necessary that the capillary tube have exact amount of blood level. In cases if there is no filled capillary tube to balance, we can use an empty capillary tube. Capillary centrifuge at 13,000 plus or minus 2,000 rpm. Remove the capillary tube from the centrifuge. It will show 3 layers. The top layer is of plasma or serum. The middle layer is thin, creamy white in color and it is known as a buffy coat. It is a layer of WBC. The last layer is the column of RBC. Use the HCT reader for finding the value of HCT. Cord reader: the reader is used as hold the tube against a scale so that the bottom of the red cell is matched with the zero line of the cord. Move the tube across the cord until the uppermost line of the plasma reaches to 100% line of the cord. Check to make sure that the bottom of the red cell column is still in the line of 0 and the tube should be straight and vertical. The line that passes through the top of the column of RBC gives the HCT value. importance. The decrease in PCV is a suitable measure of anemia. The fall in HCT may seen in decreased oxygen supply in the blood cells, heart diseases or malignant condition. The HCT also rises in case of dehydration.